The skeletal system is the structural framework of the body composed of approximately 200 bones in adults. The bones have an inner trabecular layer composed of cancellous bone and an outer cortical layer composed of compact bone. Bones are composed of 4 types of cells, namely osteoblasts, osteocytes, osteoclasts, and lining cells. The formation of the bone is carried out by active osteoblasts. Osteoblasts synthesize type 1 pro-collagen molecule into the ECM, later forming collagen matrix. They also synthesize osteocalcine and osteonectin that bind to collagen and to hydroxyapatite crystals, a crystalline mineral of the bone. Calcium is then deposited in the form of calcium phosphate, while hydroxide and bicarbonate are added and the hydroxyapatite crystals are formed. As the bone matrix is formed, osteoblasts cease synthetic activity and become osteocytes embedded in the bone matrix. These osteocytes are interconnected via canaliculi, which allow transfer of calcium ions from the interior of bone to surface, in the process called osteocytic osteolysis. Bone resorption, on the other hand, is carried out by osteoclasts, which are multinucleated cells that resorb calcified bone. Osteoclasts not only extract the calcium from the bones, but also destroy the bone matrix, resulting to a decrease in bone mass. Osteoclasts attach to the bone using integrins that bind to vitronectin found on the surface of the bone matrix. This attachment creates a lacuna between the ruffled border of the osteoclast and the surface of the bone. Osteoclasts contain carbonic anhydrase inside the cell, which provides the cell with high intracellular hydrogen concentration. The high hydrogen concentration is used by the osteoclasts in order to secrete acids and proteases via the V-type hydrogen pump into the lacuna. The carbonic anhydrase also forms bicarbonate inside the cell, which is removed via chloride bicarbonate exchangers, which are found on the membrane opposite the side of the lacuna. The acidic environment in the lacuna dissolves the bone mineral, while the acid proteases hydrolyze the matrix proteins, destroying the bone matrix. After resorbing some of the bone, osteoblasts replace osteoclasts in order to promote new bone growth and mineralization on the same site. Parathyroid hormones indirectly stimulate bone resorption by binding to receptors on osteoblasts, which stimulates the release of IL-6 and rank ligand, which promote resorption. The rank ligand binds to the rank receptor on the osteoclasts, stimulating bone resorption. Bone resorption is also stimulated by parathyroid hormone and vitamin D. These two factors stimulate osteoblasts to release MCSF, which promote proliferation of osteoclasts. Osteoblasts also have a regulatory mechanism which protects bone from resorption. Osteoblasts secrete osteoprotegerin, which when bound with rank ligand, protects the bone from osteoclastic activity. Calcium is a major component of bones. It serves a multitude of functions like the creation and maintenance of action potential, muscular contraction, motility, cytoskeletal rearrangements, cell division, cellular secretion, and in modulation of enzymes. Calcium in the body may be classified as skeletal or non-skeletal. Skeletal calcium comprises the bulk of the body calcium store, while the non-skeletal calcium may be classified into either free ionized calcium, protein-bound calcium, or calcium complex with anions. There are several factors that regulate calcium in the body. They are mediated by a calcium-sensing plasma membrane receptor of G protein link type and are located in cells producing parathyroid hormone, vitamin D, calcitonin. Bones, renal tubular calcium reabsorbing cells, and intestinal calcium absorbing cells. Parathyroid hormone is secreted by the chief cells of the parathyroid gland. Parathyroid hormone is secreted when there are low levels of plasma calcium, therefore stimulating bone resorption and tubular reabsorption of calcium. This results in an increase in calcium levels and inhibits parathyroid hormone activity in a classical negative feedback mechanism. Vitamin D promotes intestinal absorption of calcium against a concentration gradient, resulting to an increased plasma calcium. Calcitonin, secreted by the parafollicular cells or C cells of the thyroid gland, inhibits renal tubular absorption of calcium. This is stimulated by vitamin D3 and ingestion of food. During states of calcium and phosphate imbalance, hormones regulate plasma levels of these minerals. During hypocalcemia, wherein calcium levels are low, there will be an increase in parathyroid hormone secretion, while there will be a decrease in calcitonin secretion. However, during hypophosphatemia, parathyroid secretion will decrease and calcitonin secretion will increase. Patients with hypercalcemia manifest neurologic symptoms such as mild drowsiness and weakness, depression, lethargy, and coma. Gastrointestinal problems such as constipation, nausea, vomiting, anorexia, and peptic ulcer disease, nephrolithiasis, and nephrocalcinosis. On the other hand, patients with hypocalcemia manifest neuromuscular irritability, paresthesia, muscle cramps, tetany, seizures, arrhythmia, and heart block.